and thanks to all of you for coming this morning. It's a great way to start off a weekend for me, so I really appreciate you showing up. Give me a show of hands of how many of you guys might be contemplating a career in medicine. Terrific. Okay, so you guys then know what a silly name personalized medicine is, right? I mean, do you go to your doctor and say, well, let's see, you've got a problem, but it's Tuesday, and Tuesday I usually prescribe this? Of course not. All medicine is personalized medicine, right? At least we hope it is. But the personalized medicine that we'll talk about today is the personalized medicine that is inspired by variation in the iconic structure of your DNA molecule. And what I'll tell you about today is the progress that's been made about understanding how each of our genome sequences, made up of the chemistry of life, the adenine, thymine, guanine, and cytosine and DNA, varies a bit from each of us, among each of us, and then that will, I hope, be a foundation for actually being able to produce a kind of a medicine that really is tailored to our individual needs. So that's going to be one of the take-home messages today. Now, our story today begins around 2001 with the completion of the draft sequence of the human genome. Now, this was a landmark in science and in biology in particular, and for a cost of about $1.5 billion, and I'd like you to remember that number for about 4 minutes, scientists from around the world collaborated together to produce a really atrocious version of the human gene sequence. It was full of holes, there was some misassemblies, this or that, but nevertheless it was fantastically useful because it was the infrastructure, the bulletin board that allowed us to connect discoveries in one field to discoveries in another field. And it was really fantastic, and we all got excited about what the future was going to be. But of course, the popular press, being the popular press, usually gets most things wrong, and even before these publications came out, Time magazine, as it is wont to do, jumped the gun and overpromised everything, because for only $3.50 you could learn about the drugs of your future, the amazing new medicines based upon your DNA and what it would mean for your life. Well, that was 2001. It's been almost a decade, but we're finally starting to see the impact of personalized medicine based upon your particular gene variants, and that's where we're going to go today. Now, let's take a view of humanity, and I like Da Vinci's picture here on the left, and I like to think of the soma, the corpus, the body, as sort of the hardware in which our genome is the software, the operating system that causes each of us to be able to be a human being as opposed to a turnip, okay? And so what we see here is at a very high level of magnification relative to this, but a very low magnification relative to the genome, is a picture of the genome of an individual man. Right? Now, you will notice and remember that your genome comes packaged into chromosomes, and you get 2 copies of each, one from mom and one from dad, and these chromosomes contain all of our genome. Now, the reason I say this is the genome of a man is evident down in the lower right-hand corner, where you can see one version of the glorious, rich with genes and biology, resplendent X chromosome, of which women are blessed with 2, and this stubby little runt, this pathetic excuse of a chromosome that makes males males. So, guys, if you have a problem with that, get over it. It's not going to change in our lifetime, okay? Now, one of the challenges in thinking about genome is understanding the sense of scale. Now, how much DNA is in that genome? Well, the answer is it's about 7 picograms of DNA. Okay? Now, how much is that? That's a little hard to imagine. Well, a penny weighs about 2.5 grams. Okay? So and that's not very much, right? And a microgram is a millionth of not very much. And a picogram is a millionth of not very much. So it really isn't very much, okay? Now, if we were to take all of the DNA molecules in one of your cells and stretch them end to end, it would be about this high from the ground, okay? A couple meters. Now, that also doesn't really give you a sense of scale, but let's think about the following. You have 10 to the 14th cells. Now, those of you that are blessed with the capacity for multiplying with scientific notation can multiply those numbers together, and what you would find is that if I were to take the DNA out of any one of you and stretch all of the molecules end to end, it would reach from the surface of the sun beyond Uranus, okay? So if you're looking at yourself in the mirror some morning and feeling kind of special, you are. That's impressive, okay? But let's really drill in and look at what our genome sequence looks like. And I'll show you a piece of the human genome sequence. Right? So here are 1,000 bases, bits of information from our genome. 1,000 A's, T's, G's, and C's, referring to the adenine, thymine, cytosine, and guanine. Now, your genome consists of 3 billion bases, so that consists of approximately 3 million pages with this much information on it, right? And if that were stacked, you can think about how thick 2 reams of paper is. Well, if you were to stack that much paper on top of each other, your genome would consist of a stack of paper approximately 60 stories tall. And reading it, which would be barely possible, at least there's one vowel, but reading it would be as exciting as reading the Los Angeles phone book 1,000 times in a row. That's not very interesting. Now, medicine so far does not really act on genes. Medicine acts on proteins. So let's explore for a little bit what we know about the relationship between genes and proteins. Okay, so there is the sequence of one gene which makes one protein that is involved in disease. This is the gene for the enzyme called cystathionine beta synthase. If you have mutations in this gene, you have a condition called homocystinemia, and it's a condition that you don't want to have. Now, how do we learn about the protein from that gene sequence? Well, it turns out about 3 billion years ago or so, something remarkable happened, which was the evolution of the universal genetic code. The language of DNA in all organisms uses the same code to relate gene sequence to protein sequence. So if we were to take the 3 letters 3 at a time throughout this gene sequence and use them in this code, we could deduce what the sequence of amino acids is in that particular protein. Because you'll recall that proteins are written in the language of amino acids, of which there's 20. So let's pause for a second and just sort of focus on the fact that languages come in different flavors. DNA is written in 4 letters, Hawaiian is in 12, and protein is in 20. So if we were to take that table and that gene sequence and decode it, this is the sequence in the language of protein of cystathionine beta synthase. So that's just a string of amino acids in a row. Now, proteins, of course, have special structures, and that structure is part and parcel of their function. Now, there is not a single person on this planet, nor even a collection of computers that could figure out what the structure of that protein would be once it's folded up. But one of the incredibly impressive computations that each protein can do on its own is to fold itself into the proper 3-dimensional structure, spontaneously, with no help. And that's what the atomic structure of the cystathionine beta synthase protein would look like. Okay? And it even has a cute little vitamin B6 sitting right there in its mouth. Okay. So that's the whole of molecular biology's central dogma. We've gone through it. God knows why it takes a semester to teach it at Berkeley, but you now are on top of it. So let's step 6 years forward from the completion of the draft human genome sequence, and I'll introduce you to the first 2 people to have their individual genome sequences obtained. The individual on the right is the famous Craig Venter, who at the time was the head of the company called Celera Genomics, which was a company which raced with the public efforts to sequence the human genome sequence. The individual on the left is the famous Jim Watson of Watson and Crick fame, one of the co-discoverers of the structure of DNA. And as a former leader of the Human Genome Project, once it was possible to sequence individual genomes, the people who did that sequenced Jim's genome for him and gave it to him right there on his own little hard drive. Now, Jim's genome sequence cost about $1 million to obtain. We don't know how much Craig's cost because he ran the company and probably got a pretty good discount. But think about that. From 2001, it was $1.5 billion to get a lousy draft. And 6-7 years later, for 1,000th less money, we've got personal genome sequences. So that's pretty impressive progress. But now, let me show you what happened in 2009. We have the Personal Genome Project 10. This is a project out of Harvard University, led by this person, Professor George Church, who arranged for the sequencing of these 10 individuals, including various luminaries like Steve Pinker and Esther Dyson of psychology and computing fame, respectively. These heroes had their genome sequenced for about $60,000 each and made their genome sequences publicly available. So if you go to the Personal Genome Project website, you can peek inside Steve Pinker's genes, so to speak, and find out what makes him tick. Now, why did they do that? With all this concern about genetic privacy, why did these heroes do it? Well, what they understood is that we need to have access to complete genome sequence information to drive the development of the technology and the tools and the computational methods that will be important for understanding what genome sequence variation is all about. So what's happening now, more recently? Well, there's the 1,000 Genomes project. And the goal here is to complete the sequence of 1,000 human genomes. This is going on right now from people of different races and ethnicities around the world. Two of the leading institutions in this are the Beijing Genome Institute, shown here, and the Broad Institute between Harvard and MIT, shown here. And this project is well underway. All of this information is immediately deposited into the public domain so that scientists around the world can use this information to drive their own science. Now I'm going to show you an instrument that is used to sequence, and it's from one company. I want to emphasize that I have no commercial relationships with any company that I'm going to mention today, and I'll even give you one piece of data about why you might not want to have any relationship with some of these companies, at least as an investor, and we'll get to that in a second. So I'll show you the Illumina HiSeq machine, which has the remarkable capacity of sequencing in one machine, in 1 year, the genomes of 104 people. So there it is. Looks like a European dishwasher, right? But inside it's pretty darn impressive and uses the latest applications of both physics, optics, and chemistry to do this remarkable thing. So the Beijing Genome Institute has about 128 of these things. They're on the order of $1 million a piece. The Broad Institute has 50. As of the first quarter of this year, those two institutions had 200 of them on back order. So we expect then, next year, 500 of these machines will be in the field churning away at genome sequences. Now I'm going to extrapolate. And extrapolations are always dangerous. But I'm going to extrapolate for improvements in this technology on this machine for the next 2 years that are smaller than the advancements that were made for the last 2 years. So I think this is a conservative extrapolation. So if we do that, and we assume that the increase in capacity that will be about 5-fold in 2012 and 4-fold in 2013, then we get to the shocking realization that while you're in college, this machine brand alone will sequence 1 million human genome sequences. Okay? So 9 years ago, 1.5 billion to get a lousy version of one. A little while, we're going to have 1 million from this one company alone. Now, if you think that's something, within 50 miles of where we're sitting, these companies are working feverishly to develop machines that'll blow the doors off the one that I just showed you. So there's no question that many of you, and possibly I think all of you, will have your entire genome sequenced for not very much money in the not very distant future. So what we'll do today is to explore what you might be able to learn from that. So I think this is a very exciting time. Now, why I said you might not want to invest in some of this technology in terms of an investment is shown on this next slide, where I discuss the plummeting cost of sequencing, which is producing this genetic fire hose of information. So here's a graph that you've probably seen part of before. Now notice on the y-axis, so this is a logarithmic scale. So the line here is the famous Moore's law that my electrical engineering colleagues have been lording over me for years about how impressive the advancements in computing are, so that every year or so it doubles the capacity and decreases the cost by half, right? Well, this curve represents the incredible increase in efficiency of DNA sequencing. Now, that's impressive. Now, if we use the methods that were pioneered by Goldman Sachs and extrapolate, what we see is that the week after next, sequencing is going to be free, and halfway through next year, companies are going to be paying you for the privilege of sequencing your genome, assuming nothing changes before then. Okay? So this is technologies advance beyond belief. Never in my career have I seen advancements like this before, and I see no sign that the acceleration is not going to continue. So this is really astonishing. It brings us to an incredible age. And this age is represented by B. Reinhoff right here. B is the poster child for personalized medicine. This is her father, Hugh, who has been working to solve a fundamental medical mystery. B was born with a medical condition in her connective tissue that is possibly unique on the planet. It's never been seen before. It has baffled all of the experts. And Hugh decided that he wants to search for the misspelling in her genome that is affecting the health of his daughter. Can you imagine that, right? Well, Hugh's a pretty enterprising guy. He put together a website, My Daughter's DNA, to help others who are in this situation. And Hugh estimates that there's probably something in the order of 6,000 different rare medical conditions for which the underlying genes have not been found. And to give you a sense of how rapid progress can be, let me share with you a recent email that I received from Hugh. So let's check this out. Saturday, December 4th. Pretty recent, right? 4:30 in the morning. Yeah. Dear Jasper, you might be interested to know I found B's mutation using exome sequencing of the family. As predicted, affecting TGF-beta signaling. Incredible technology we should all have access to. Best wishes, Hugh. Can you imagine what that must feel like to solve the medical mystery of a disease affecting somebody in your family? Wow. So let's go back to our genome sequence, right? Page 1 of our 3 million pages of awful A's, G's, T's, and C's. How do we make sense of that? Well, what we make sense of is not by studying it all, but by looking for the variation. Here's an example of the kind of variation that I'll talk about today. This is called a single nucleotide polymorphism to represent that this particular base pair, this GC, is converted to this AT. There's a language that's used to describe these. They're called SNPs, SNPs, for single nucleotide polymorphisms, mostly because scientists are lousy typists, and SNPs is easier than single nucleotide polymorphisms. So there are many other kinds of variants in the genome, but this is the most common, and this is the kind that I think will be really the target of the personalized medicine that we were talking about earlier. So what do we know about those? Well, we know that they occur at a frequency of about 10 to the minus 8th changes, we'll call those mutations, per base pair per generation. Okay? So per base pair, it's pretty rare. But there are 7 times 10 to the 9th people on this planet. So if you do the simple arithmetic, you will come up with the astonishing insight that in each generation, all possible simple mutations occur at all positions in our genome. Okay? So there is nothing that you can learn about any gene variant that isn't really relevant to somebody. Some of these variants are very common. They'll be shared by 50% of the people on the planet. And others are relatively rare. You might find them only in one person in Ulaanbaatar, but all of them exist in somebody and are recreated each generation. So that's pretty cool. So now we're going to explore what we know or what we're starting to learn about genetic variation. And if you're going to explore a strange new world, you really want to have a guide, something like a Lonely Planet guide where you to go to deepest, darkest New York City, right? Well, there is a guide that's been written for the human genome. It's called A Guide to the Human Genome. This is sort of not at the high school level, it's at the advanced undergraduate level, but let's ignore the guide. What we really need are 4 principles to guide our thinking. And these 4 principles are ones that you've probably encountered, and there's only 4. So the first one, of course, is the principle of evolution by natural selection. As Dobzhansky is said to have said, nothing makes sense in biology except in the light of evolution. And Darwin and Wallace were the originators of our modern synthesis of evolution. Now, people, we accept evolution so broadly that it kind of becomes blasé, but let me really drive this point home by a discussion of the so-called Werner's gene, abbreviated WRN, and I call this the ageless aging gene. So if we look at the sequence of that gene, written in the language, in the chemistry of proteins, here's the sequence of part of it from human. Now, if we were to look at that gene in the genome of the dog, mouse, chicken, and fish, we would see it's the same. Nothing has changed. Okay? But if we look a little more divergently in evolution at the worm, fly, yeast, rice, and yeast, we see there are some changes, but more or less the gene is mostly unchanged. Okay? So the evolutionary clock ticks at a fairly slow rate. So what does this gene do? Well, if we go into this gene and change that amino acid right there in yeast, what happens is that yeast, the creature that makes bread, dies prematurely. It doesn't live as long. Now, if we made that same mutation in a human gene, here's what happens. That's a 6-year-old boy with the physiology of an 80-year-old man who will die shortly. Okay? So this is an amazing demonstration, really, that Darwin and Wallace were right beyond their wildest imagination, that the evolution has happened, the conservation of function is conserved from yeast to human is something as important as the fundamental process of aging. So that's one principle, and it's a good one. The other principle that's very useful is just a knowledge of human anthropology and human evolution. What we now recognize beyond any doubt at all is that humanity of all types originated in Africa. And humans first started leaving Africa to populate the rest of the planet about 50,000 years ago. Now, at the time that they did that, the population in Africa was on the order of 10,000 people, but the groups that left were small groups that left at different times. Now, if you have a very large bag of marbles of a fixed ratio of different colors and you were to reach in and grab a few, the sample that you would take would not represent necessarily the distribution of the colors in the original bag. The same thing is true of genes. Africa is rich, rich, rich in genetic diversity, richer than any other place on the planet. And each group that left Africa took a different selection of gene variants, I'll call them alleles, with them when they left. So by scanning a person's genome, you can actually figure out, because of the frequency of different variants, which part of the world their ancestors were likely to come from. So gene variant frequencies vary very greatly between different populations. So that's a principle. The third principle is human pedigree analysis. So you are all familiar with, from biology, these kinds of diagrams that represent a grandfather, a grandmother, their 5 siblings and spouses, and their grandchildren. So what I'm showing you here is the inheritance of a trait, redness, right? Grandfather had a red trait, some of his children had that trait, and some of the grandchildren had that trait. Well, you know that the genetic deck gets shuffled in each generation in meiosis. And so this son has half of the genes from his father, this granddaughter has 1/4 of the genes of the grandfather, and you can imagine how if you sampled across the genomes of this individual, that individual, and that individual, each of which share 1/4 of the gene variants in the grandfather, that with a reasonably sized family, you could pretty much zero in on where the gene for redness might be. And that's a conventional method that's been used to discover many, many important disease genes. And the last principle is a very interesting one, and it's the most misunderstood one in the press. And this is called guilt by association, and it says commonly practiced, it's referred to as a genome-wide association analysis. So the principle here is beguilingly simple. Let's think about the following. Suppose you're interested in finding genes responsible for a particular disease, so the disease du jour. So what you would do would be to collect a bunch of people that share that disease. Then you would collect a bunch of other people that don't have that disease, they're healthy, and they would be your control group. Okay? Then what you do is to sample variants known across the genome, common variants, and you simply ask which variants are more common in those with the disease and are uncommon in the healthy individuals. And as you can see here, at this particular gene, the A version at that position is more common in the healthy individuals, whereas the G version at that same position is much more common among the diseased people. Okay? So what this would let you say, if you had large enough numbers to provide statistical rigor, as an aside, if there was one piece of advice I could give any student thinking about going into science, it's to get a really good foundation in statistics. We are in the information age, and statistics is the science of data. So, you know, that was the most useful course I ever took. Okay? Back on track. So, is this gene variant the cause of the disease? Maybe. More often than not, not. But if the statistical evidence is strong enough, you can say that this is an association of the disease with that particular gene variant. Why might it not be the cause? Well, because genes travel in packs. And the causal gene might be the one right over here, which you simply didn't sample. Now, because this point is so misrepresented in the popular press, let's do an experiment right here today, and we are going to test my hypothesis about the genetic foundation of a particular trait. I'm going to declare that haggis eating is caused by a genetic disease. Okay? So what's haggis? Well, it's disgusting. It's a Scottish dish. It's made of the guts of sheep and calves that you mix up with lard and spices, and you boil it in the stomach of the calf, right? And then you serve it up. Good. Has anybody here had haggis? Okay, so you can see it's a pretty rare disease, right? So how are we going to find the gene that would cause somebody to eat this stuff? Well, what we need is a large population. So Beijing will do, and we'll set up a booth. And this booth will say across the top, free gene testing for all haggis eaters, right? And so all the haggis eaters will come up and they'll spit in a tube and give us a copy of a saliva. And then what we'll do is to take a couple of controls that are non-haggis eaters, and we'll scan gene variants across the genome. And you know what? Without a doubt in my mind, we are going to find gene variants which are absolutely common among the haggis eaters and are hardly ever seen among the non-haggis eating controls. So do we publish that we have found a genetic association with haggis eaters? Well, before we do that, we ought to look at the properties, the phenotypes of these haggis eaters, and what you will find is that they share an unusual fashion consciousness, right? They tend to be light-skinned, freckled, red-haired, and an amazing fondness for really dissonant, loud, bizarre music played on ugly instruments, right? So have we found a gene for haggis eating? Of course not. We have found gene polymorphisms that distinguish Scots from Chinese, right? So association is interesting. It is important. It can be useful, but it is often not causal. And if you read about a genetic association, I don't take any of them as seriously until they've been replicated 3 or 4 or 5 times on different populations for reasons that are now evident to all of us. So those 4 principles then lie behind our approach to analyzing complete genome sequences. So let's think about what we've learned so far. So what we've learned from the complete sequence now of a growing number of people is that in fact there are 3 billion bases in our genomes. Fortunately, the computers have not alphabetized this on us. That would be a disaster. And between any 2 of us, there are about 3 million differences of the single nucleotide polymorphism type. Now, the parts list of humans is going to be debated for decades. The parts list of yeast is still being debated, but it is going to be approximately 22,550 proteins encoded in our genomes. That's going to be accurate within 200, I'm sure. There are, among those 3 million differences in genes for proteins such that 2,000 differences will be between any 2 of us in our proteins. Okay? So none of us are normal. Of those differences in our proteins, we can calculate now with reasonable predictive value that about 120 of those are going to be deleterious. So none of us is normal. We're all carrying around bags of mutant genes that are deleterious with respect to making proteins. And of those, 5 of them we expect to be potentially lethal if that were the only version of that gene that we carried. Okay? So it's a good thing we have 2 copies of all of our genes, and this is the reason that it's a really bad idea to mate with your brother or sister. Okay? Now, the other cool thing is that we know by the sequencing of identical twins that there are about 80 new mutations that occur in every generation, every cell generation. Okay? So variation is the stuff of humanity. It is what makes us different. It's changing at a slow frequency, but it's happening all the time, which highlights the central dilemma of personal genetic information. Now, that's not a famous human geneticist, but is a very famous human, Mark Twain, who had better comments about more things than anybody else I know. And his comment that I liked the most was that, You know, everyone complains about the weather, but nobody does anything about it. So the question is, with respect to our genes, you know, we all have problems in our genomes, but is there something we can do about it? And part of the problem has been the following. Most people think that most personal genetic information is bad news. Why is that? Because it's the bad news that is the headlines. Okay? And that is coupled with another issue, which is that learning your genetic information really has the capacity to invade somebody else's privacy. So let's imagine the following situation to illustrate this. Let's imagine that one of your grandparents has some disease that you really don't want to have, something like Alzheimer's. Now, most cases of Alzheimer's are not caused by well understood genetics, but there are some which are. So let's consider such a family and such an individual. This grandmother has Alzheimer's, okay? Now, mom is taking care of that grandmother, and it's a big task. And it is helping mom see what that future could be like if she has inherited that mutation, and frankly, mom just doesn't want to know. But suppose you, armed with a much better understanding of science, biology, and genetics, say, gosh, you know, I really want to know. Because if I know, I can do things like buy long-term health care insurance before it becomes illegal to do so based upon your genetic information, right? So if you, one of these grandchildren here, have that test done and find that you carry the version of the gene responsible for your grandmother's Alzheimer's, then unless you can keep that information secret from your mother, and we all know we have no secrets from our mother, right, then you have invaded your mother's genetic privacy. And that's a big issue, and frankly, there is no technical solution to that. It's the nature of the age. So what I want to do now is to turn the coin over, and I've shown you sort of some downsides about genetic information, but let's look for good news in our genome. And what would it look like? Well, I think it would have various qualities. I mean, importantly, it would be non-frightening. Frightening information is not good news. It should be actionable, it should be safe, it should be inexpensive to act upon, and it should be effective. And the inexpensive part means that the pharmaceutical industry, by and large, is not going to be searching for this kind of information because they're looking for the expensive information that they can make profits on. That's fine because they're the companies, they have to do that. Good news should have minimum privacy issues. It should improve your well-being. And I believe, and time will tell, but I believe that nutrition and the way it intersects with our personal genetic variants is going to be the sweet spot where we're all going to have access to personal genetic information that we can act upon. And I'll show you why. Now, this idea does not come from me. This idea comes from my dear friend and advisor of Mark, Bruce Ames, one of our colleagues here at UC Berkeley. Now, Bruce has made the world safer for all of us by discovering the mutagens in the environment and many, many other things. He's received many well-deserved prizes. He hasn't deserved the biggest prize, which he clearly deserves. He hasn't gotten the ticket to Stockholm yet. And in his possibly most obscure paper ever, published in 1971, is this fact. 20% of certain inherited diseases of bacteria are vitamin B6 remedial. Now, I read this paper in the 70s, and I said, that's cool, and then didn't think about it for about 40 years, right? Until I suddenly realized that Bruce has the answer that I'm looking for, right? If you look at it in just the right twisted way, what we have here are inherited diseases of bacteria that can be treated by something as cheap, safe, and effective as a vitamin, okay? So is there anything special about bacteria? Could the same thing be true for humans? Well, let's explore biology to see if that could be true. So what do you think these animals all have in common? All primates, the guinea pig, and the ever adorable fruit bat. What's in common with these animals? Anybody know? They're mammals, but there's other mammals too. Only these animals share one trait of relevance to our discussion today. Okay, did anybody give your dog or cat a vitamin C tablet this morning? No. And the reason you didn't have to is that your dog and cat and every other animal except those at the top can take glucose and turn it into vitamin C. Vitamin C is incredibly important for lots of things, scurvy being among the least of them. Okay? But all those animals on the top have mutations in the gene that makes this enzyme, right? So we all share that mutation. Now, we don't think of that as being a mutation. We think of that as being normal because we all have it. But in fact, with respect to biology, with mammalogy, we're all mutants. So could this be the only such example in the world? Well, I think not. Right, here's a very simplified version of the biochemical wiring diagram of metabolism. So each dot here represents a chemical entity of the biochemical transformations that happen in each of our cells. The lines represent enzymes that convert one dot to the other dot to make things, like amino acids, DNA, proteins, hair, stuff like that. So there's about 10,000 different reactions in human metabolism. Each use an enzyme catalyst, and there are 640 enzymes that require, for their function, a vitamin or a mineral bound to that enzyme, like that B6 molecule that I showed you in the mouth of cystathionine beta synthase, for that chemistry to happen. So Bruce has written powerfully and persuasively that variation of these genes might really be underlying lots of our differences in our metabolism, and for the last few years my lab has been describing such variants, and frankly there are lots of them. And I think we're going to learn a lot about our own individual nutritional requirements because our RDAs for vitamins and minerals were based upon science done in the 1940s. And very little change has happened since then. So it's amazing to me to think that the science that we all depend upon for our daily life is ancient. Okay? So what about you? How could you go about learning about your personal genetics? Has anybody in here been tested for, had any genetic tests done on you? Just a couple? In fact, you all have. Okay? If you were born in this state or any other state, and most nations, within a day or two after you were born, blood was drawn from your heel and put onto a card called a Guthrie card. These cards are stored by public health departments. And your blood spot was tested by methods not as reliable as direct DNA measurements, but they're gene tests nonetheless, for these inherited disorders. Why? Because these disorders would have grave consequences to you if something wasn't done, like changing your diet, to protect the grave consequences of these. Okay? So you are all subjects of genetic tests already. Now let's take a poll. Suppose I would ask you the following question. Should all college or high school students be subjected to mandatory, non-anonymous gene testing for serious medical conditions? What do you think? You think yes? No. How many no's? Okay, me too. But it's already happened. As of this April, the NCAA has mandated gene testing of all Division I college athletes at least for the sickle cell trait. Why? Because carriers of the sickle cell trait over the last couple of years in 8 instances have dropped dead after heavy workouts for sports teams. Right? So in a sense, this is a good thing. It protects the health of college athletes against very significant outcomes. But it's mandatory and it's not anonymous. Okay? If you get let off the team following a gene test, people know why. So that's interesting, and that kind of brings us to this interesting age of direct-to-consumer gene testing, which has really created an enormous amount of controversy recently. And what we see in this particular situation is the Food and Drug Administration, the American Medical Association, and legislatures are sort of sparring off against the interests of entrepreneurs, consumers, and challenging really the fundamental question about who has the right to know about our individual gene sequences. And I'll show you a dramatic example of that. And this is a very unfortunate situation where technology and knowledge gets way ahead of our understanding. Because what we can do as a society depends not upon what you and I know, but upon what our society knows collectively. And I'll show you how that manifests itself interestingly. Now, to prepare for this and other related discussions, I decided to have my DNA profiled by one of the local companies. This is one that Francis Collins, the director of the National Institute of Health, has also had his DNA profiled by. And by and large, he has come to the conclusion, based upon comparing the efficacy of various different companies, that the leaders of this technology produce reliable data. The technology is really robust. The interpretation of that information is open to question sometimes, but the technology is robust. So let's take a look inside my genes and see what I'm at or disease risk for, okay? So what we're looking at here are conditions that I have an elevated risk for, the average risk of the population, my relative risk, the confidence of this company in making this assessment, and some quantitation over here. So let's really talk about these numbers. Which numbers are solid and which ones are less solid? Population-wide risk averages are pretty solid. Confidence. A quantitative argument by stars, you saw that in elementary school. Gold star, 2 gold stars. What's that all about? Well, what it's all about is a mix of science and art. It is an expert interpreting the often conflicting published literature, using as much information as they have available to them to make an educated guess about what the current state of the knowledge is. So it is imperfect, but it is good. Okay? Not great, good. So what's in store for me? Well, I was surprised to find out that I consider most of this good news. Okay? So psoriasis. I am at a 3 or 3.8-fold higher risk for psoriasis. Now this does not bother me a great deal, but you know, certain times of the year the skin of my hands peel. It's kind of nice to know what that's all about. What else? Atrial fibrillation. Well, my risk is only trivially higher than the genome-wide or the population-wide risk, but it's scary to think that a heart condition has a population-wide risk of 27%. So what did I do with this information? Well, I dragged my colleague out to the gym more often so we can make sure that our hearts at least get the benefit of a more periodic workout. Macular degeneration. Okay? I have about a 1.6-fold elevated risk of macular degeneration. So you might not think that's very high, but the population-wide risk is 7%. So how do I use this information? Well, macular degeneration is a form of blindness that's common among old people. Now, you may not think this, but I am very, very far away from being old. But when I went to have my eye exam at the clinic, I told Dr. Peng, hey, you know, I'm a carrier for something which predisposes my risk for macular degeneration. He said, really? I said, really? He said, well, let me take another look. That's the important thing, right? That information had him take an extra look. Did he find anything? No. But he took an extra look, and that's a good thing, right? So this is an example of the way this information can be good. What about carriers, right? What am I a carrier for? Well, it turns out I am a carrier for something. I'm a carrier for a disruptive mutation in a gene called alpha-1 antitrypsin inhibitor. And a deficiency in this creates a problem in your lungs if you work in a dusty industry, right? So carriers are fine. But if you have 2 copies of this gene, you're going to have 2 copies of this variant, you're going to have lung problems. Interestingly, my grandfather died at about my age from lung complications. He smoked cigars his entire life, and he worked in a dusty industry. That may have been where I got my allele from. Okay? So am I at risk? Not particularly, because I have 1 good copy of that gene. Now, if you look at the rest of these conditions, nothing but gold stars, right? You think, ha-ha, I dodged the bullet. But have I? So let's take a look at a couple of these genes, and in particular, Brca1 and Brca2. Now, I'm sure that some of you have some familiarity with this. These are the genes in which mutations cause early onset breast and ovarian cancer. Now, those of you going to medical school would be confident in predicting that my risk of ovarian cancer is 0. Right? And as a male, my risk of breast cancer, even were I a carrier of one of these mutations, would be low. But I wanted to know why this company thought that I was not a carrier, so I clicked on these various links, and come to find out, it's very interesting, and they're very upfront about this. They tested my genes for the 5 most common variants of the Brca1 gene, and I don't have those 5 most common variants. However, there are over 1,000 different variants that have been described in that gene at this point that cause early onset breast cancer, and they didn't test any of them. Okay? So, if you overinterpret these data, it can lead you to a false sense of complacency. So you really have to understand the data, what the limitations are, and the reasons are both legal, because there are patents by other companies on the other variants, and technical, that the technology has not been applied genome-wide. They're just looking at common variants at this point. There's another kind of information that one can get, but I'm going to skip this for the sake of time. It's information that tells you what drugs you might be particularly sensitive to. I'm a little more sensitive to Coumadin than others. That's important. If I ever have an operation, Coumadin is a common anti-coagulation effort. Now, because of the importance of personalized genetic information, Berkeley did a very interesting experiment this semester actually, as part of our On the Same Page program. And some of you may have heard about this, some of you may have brothers or sisters that engaged in this. The idea in the past has been to give our freshman class something common to think about, to discuss with each other. And we've given them various books to read in the summer before they come to college, very interesting books. And we discovered, interestingly, that most students in the summer before they come to college don't have a lot of interest in reading very interesting books. They have other things on their mind. So we decided to try something else. And the deans of the College of Letters and Science said, hey, could you ask me if I could lead a program in personalized medicine? And I said, okay, I could do that. What do you want me to do? And I said, well, could you sequence the genomes of the entering class? I said, well, no. And they said, well, what could you do? I said, would you be happy if I tested a few common gene variants? They said, oh yeah, that's just as good. Where did they get deans from, right? So what we decided to do was to have a voluntary gene testing program for 3 gene variants that was entirely voluntary and entirely anonymous. So everybody in the entering class received in the mail an explanation of the project, a kit to spit in if they wanted to participate, and 2 anonymous barcodes and a consent form. They had to sign the consent form saying that they had read the material and had watched an informational video from our website. And they had to put 1 barcode on top of their spit kit and keep 1 barcode for themselves, because that barcode would be the only way that they would ever be able to get access to the data from their genes from their spit once we measured it. So the samples were sent into the dean's office. Staff in the dean's office took the consent form. If it was proper, they shredded the consent form and passed the saliva sample on. If it was not a proper consent form, the consent form and the saliva were destroyed. So we wanted to give our students access to the experience of deciding whether they wanted to have personal genetic information and some genetic information. The genes we chose, with only 3, anybody would choose different genes, no doubt. My thinking was the following. I wanted people to have a gene test for a condition that they probably already knew about themselves, another test for a condition that they were likely to know about themselves, and another test for a condition for which the scientific literature had been controversial but had been resolved so that people could understand how confusing it can be sometimes. So the program was launched with big fanfare here at Berkeley, and it got a certain amount of attention, including here in the New York Times. And I'd like you to note the date on this article, May 18th, 2010. And Tamara Lewin wrote a largely favorable and largely accurate article about what it was that we were trying to do. Now this got a lot of attention, including the attention of Russ Altman, who is the smartest professor at Stanford. Now, Stanford has a lot of smart professors, so how do I know that Russ is the smartest? Well, look at what he wrote. I can't believe I'm saying this, go Cal. Here's what he wrote. He says, I believe it is absolutely critical that we immediately start educating students about genetics and the use of genetic tests in making decisions in life. I think that direct engagement with personal data is one very effective way to make all the issues crystal clear. Now, feel his pain as he typed the last line. So, at least in the narrow arena of providing genetic information so students can learn, go Cal. We have finally found a Stanford professor smart enough to recruit to Berkeley. Okay, so what were the 3 genes that we chose? One gene was the gene for lactase and the variant that causes lactase to be continuously produced in adults. So lactase you need to digest lactose, which is the sugar in milk. And most people on the planet lose the ability to digest lactose as they become adults because this gene turns off. And it's only those of us who are descendants of Northern European herders who have a change in the gene up here in the switch region of the gene that locks that gene in the on state. So we reasoned that by the time students got to Berkeley, they pretty much knew whether or not a milkshake made them nauseous or not. Right? So that was the one that we thought they might already know about. And this is really interesting and common and widely studied gene variant. Another gene variant that we studied, which was surprisingly controversial, was the gene variant in this gene here, aldehyde dehydrogenase. Now, we reasoned that none of our students would have yet drunk alcohol before they got to Berkeley, but when they did, the enzymes in their body, alcohol dehydrogenase, would convert ethanol to acetaldehyde, and then this gene encodes an enzyme that degrades acetaldehyde to acetate. Now, variants in this gene are very common in people of Asian origin, and these gene variants are responsible for the flushing that happens often when Asians drink alcohol. So we thought that this would be a variant that would help them understand what was going on when they had their first alcoholic beverage in their senior year. And the other variant that we looked at had to do with mother's advice to eat your vegetables, eat your leafy greens. Your leafy greens are the source of folic acid, and folic acid is a crucial nutrient, and there is a common variant in this gene, methylene tetrahydrofolate reductase, which is shared by approximately 150 people in this audience, and 30 of you have 2 copies of this particular gene variant. Okay? I'll tell you more about that in just a second. So this variant, which goes by the colorful name C677T, converts a cytosine to a thymine. In the literature, over the years, multiple studies showed had an association. Right? We know about associations now, not causality, an association with significant effects, birth defects and coronary disease. But larger studies that are more recent have not been able to reproduce any of those. And so this is a common example in the medical literature. We want our students to understand that. I mean, how do you know what you can trust? Well, I have to remind people from time to time that the standard for publishing something means that you are 95% sure that the result you got did not occur by chance. You have to be at least 95% sure. That's the standard for publication, which means that 5% of the time you're going to be wrong. There have been 1,000 papers published on this variant. Guess what? Some were wrong. The more modern studies have overturned that. So we proposed to do this, although we didn't have to. We had this entire project reviewed by the committee on the campus that protects human subjects that are participants in research. This was not a research project. This was an educational project. We had it reviewed and approved at all levels. When we got our samples in, we got 726 properly consented saliva samples. They were scanned and followed by its barcode. That's the only mark that we knew about them. Fancy machines were used to measure gene variants in those samples. We measured gene variants using this technology from Illumina that I'll not spend the time to go through, but suffice it to say it's really terrific. It uses very expensive gizmos to get the data out of it. And we really took a lot of effort to protect the privacy of our students by, first of all, there was only a barcode ever on the sample. We removed the barcodes when we were done. We destroyed the DNA by soaking it first in bleach. Then we packaged it all up and sent it to an incinerator in San Leandro and put in all kinds of controls beyond belief, so that we were sure that we had extremely high quality data on 724 of those samples. 2 we did not. And I would have been seriously disappointed in the entering class if at least a couple of them didn't get their dog to spit in the saliva tube just to test us. In fact, I found out later that one of them, in fact, was dog saliva. You know, I love Berkeley students. They're keeping us on our toes all the time. So what did we learn? Well, what we learned is that the Berkeley population is much like the world's population. We measured the 3 different genotypes, the CC variant, the CT heterozygotes, and the TT homozygotes for the MTHFR variant, the lactase variant, and the aldehyde dehydrogenase. And I was very excited because we were going to be able to give students, for the first time ever at any campus on this scale, a peek into their genes, personal genetic information. But it turns out this project that all of the deans and I thought was a great project and a great idea received a little criticism. Okay? Note the date, May 19th, 2010. 24 hours later, the critics have come out of the woodwork. And we had approximately 200 editorials written in the major papers around the country, some being like Professor Altman saying, this is terrific, we all need to get to this level of understanding. The GDP, 17% of our GDP is spent on healthcare, we need to do this better. The other half saying the world is going to come to an end because they're testing Berkeley undergraduates for 3 gene variants. It was amazing. But most amazing of all was the following. Whoops. Last summer, an assemblyman from Orange County introduced Assembly Bill 70, whose goal was to make this project illegal and to fine the university for the amount of money that we spent on it, taking it out of the general fund. Wow, who knew? They were so tuned in. So I had a chance to spend some of my summer vacation in Sacramento. I got to know this man very well. Steve Juarez, he's Associate Vice President for UC Government Relations. Steve and I went to visit that assemblyman to try to explain to him why this was, so benign and so good and so interesting. That went nowhere. Then we had a chance to testify at the assembly, hearing at the assembly. That was very interesting. And finally, we had a chance to testify at a hearing at the Senate. And at the end of that hearing, the vote was to kill that bill and to let the project go forward. And boy, I'll tell you, we were ecstatic for 45 minutes. Yeah, I know. And the reason is that we had received a note from the California Department of Public Health advising us on not to go forward with this without explaining it to them. So we went to explain it to them. Now, why were they involved? Well, in 1988, there was a law passed called the Clinical Laboratory Improvement Amendment, which was a very good idea. And essentially what this law says is that if you're going to do medical tests, you have to do these tests in a CLIA-certified lab, which means it has proper establishment of controls, and there's physicians involved in the process, and this or that. It's kind of complicated. And I don't think there's any CLIA-certified lab on this or any other UC campus. Possibly UCSF has one, but I'm not sure about that. Now, we were worried because there is an explicit exemption written into the California Professional Code, section 1241, subsection B, subsection 4, which completely covers us for this project. And let me explain it to you, which is, so this law exempts from regulation laboratories or personnel that perform clinical laboratory tests or examinations for research and teaching purposes only. That's UC Berkeley, research and teaching. And does not report or use patient-specific results. We got no patients, we got students, right? For the diagnosis, prevention, or treatment of any disease or impairment of, or for the assessment of health of an individual. We're not doing that either. We're assuming all of our students are healthy. Now, if you ever find yourself as a scientist interpreting a law, you're doing the wrong thing, right? Because we don't know the law, but these guys do. So this is my dream team. I had the lead counsel for the University of California Office of the President. I had the lead counsel from UC Berkeley's office, and I had another lawyer who's the Assistant Vice Chancellor for Research Administration here. All checked this out. They all said, yeah, man, you are completely covered. So a couple of these went to my audience with the Director of the Department of Public Health, Dr. Mark Horton, and we explained why this was completely benign. And he said, well, this is, you really couldn't even measure hair color. I said, really? He said, yeah, that's giving medical information because redheads are more prone to melanoma. So at the end of the day, the director said no, that we could not give the students their own genetic results. Now, I was as baffled by that decision as this gentleman seems to have been on a different occasion with respect to something that Dr. Horton was saying. And all I can say is that the director of the Department of Public Health thought it was too big of a risk somehow that we would give UC Berkeley students information that might guide their consumption of ethanol, milk, and their leafy greens. So, is there going to be a personalized medicine? Well, there are certainly some obstacles. One is the fundamental question of who should have access to your genome. Dr. Horton's view was that a physician has to be involved in ordering any genetic test and in receiving and interpreting any genetic information for you. I went to my own personal physician with my gene profile. I said, would you like to look at this? She said, God, no. My colleague here who got her MD at Stanford took not a single genetic course in her entire MD training. So why is it that physicians are supposed to be involved in the loop? How do we keep our information private if some other person has to be involved in the loop? There's lots of other issues, including education, issues involving the clinical trials, FDA approval, and of course privacy. But I will say one really important thing happened in the Bush administration, one good important thing, and that was the passage of GINA, the Genetic Information Non-Discrimination Act. And I think this is a really important first step. It protects all of us against discrimination in employment and in health insurance. It excludes life insurance. It excludes long-term health care. It's not perfect, but it's a start. So what do we see about the future? Well, predictions about the future are always dangerous, right? And you got to get the details right. But I see this personalized information having untold numbers of different applications that are really going to fundamentally change the landscape of medicine. And I'll show you one local example. Here is the sequence of the human gene for CCR5. This is a protein that sits on the surface of immune cells, and this is the door through which HIV infects cells. Now, 11% of Europeans have a mutation in that gene. That does not completely keep HIV out, but it's pretty darn good. So think about that. If you were the lucky inheritor of 2 copies of that defective gene, you would be relatively HIV resistant, but you would be extremely foolish to think that that was an excuse for unsafe sex. But what can you do with that information? Well, a local company, Sangamo, and a team led by this fellow, Fyodor Urnov, who is the blue and gold subunit of that particular company. He teaches here at Berkeley. He's one of our very best teachers. Has been working on enzymes represented here called zinc finger nucleases. These are enzymes that allow us to go in and edit genes at will in any organism on the planet. So here's what they've done. They've given us the capacity to start to think about the C word. Can we think about cures for people infected with HIV? So what they've done is to establish that they can knock out the CCR5 gene in immune cells, they being in yellow, Berkeley undergraduates, and in one case graduate students, who worked as interns at this company. And most recently, they have shown that they can do this in hematopoietic stem cells. And there are people in clinical trials right now in which their own hematopoietic stem cells have been edited to remove the CCR5 gene. And the hope is that in those infected individuals, their immune system can be repopulated by cells that are resistant to HIV. So how would it feel to carve your initials into a discovery like that? Huh? Pretty amazing. Well, we're getting to the end. And if you want to learn more, 2 of the very best books on this subject just came out this year. One is The Language of Life by Francis Collins, the director of the National Institute of Health, and the other by my friends Stan Fields and Mark Johnston, Genetic Twists of Fate, stories about their dives into the deep end of the gene pool. I had a lot of help on this project with Lisa Barcelos in her lab, who did all of the gene testing for this program. We got a lot of help also from the Dean of the Biological Science, Mark Schlissel, who is one of the architects of the program, as well as his staff. And when an issue gets really controversial, you find out who your friends are. And leadership is an important thing. And I'm really happy to say that in the thick of it all, our chancellor, faced with hundreds of editorials calling this project and putting an end to this project, described this as Berkeley at its best. We had the help of Robert Tejan, our colleague here at Berkeley, who's also the president of the Howard Hughes Medical Institute, to fund this on completely private funds. And I'll close with Fiat Lux. Thank you for your attention.